Welcome to our virtual vaccine meeting here. It would be nice if we could all be face to face. Hopefully next year that'll be the case as we work our way through a somewhat unprecedented pandemic. I think you've already heard from, well, the first speaker who warned us a bit about how it is that we work better with our immune system and the health of the host in building future vaccines that are more effective and safe. That's clearly the challenge. I think having been mentored a bit by Maurice Hilleman and hearing his quote, of course, about a bargain basement sort of analogy for the technology, certainly vaccine technology started in very basic roots, but it's our hope that we evolve these to a very sophisticated high level of very specific and safe entities. And so I'm going to talk a bit about shifting into some of the science and technology you're going to be hearing. And that is to sort of address where it is that we might be in developing vaccine technologies for the kinds of pathogens that we are experiencing, such as this COVID, SARS-CoV-2, which I'm sure is really probably the most common used words in society today and globally in an unprecedented way. Certainly vaccines are being talked about on a daily, hourly basis. And so never before has the whole thing, I think, been put in front of people like it is today. So with that, I'm going to talk a bit about a better appreciation for what I have termed earlier in some reviews, and that is different organisms that we call class 1 and class 2 pathogens, to try to get ourselves to think a bit differently about some of those whereby we have vaccine technologies which have addressed these infections, and in other cases where they're not really as efficacious or nor have they been fully developed to protect us. That sort of weighs into the classical sort of the Jenner era-based vaccine science and technology, and some of these issues where pathogens may have evolved an immuno-dominant type of deceptive imprinting, thus that they can get around some of our developed host responses and vaccine technologies. And I'll use an example, given that we are in the midst of a respiratory pandemic, of another successful seasonal and annual virus, and that is the human rhinovirus group in the coronavirus family. And I'll then share with you how immune refocusing technology is poised to help us work through what, unfortunately, I believe will be the case with SARS-CoV-2. And that will be the unfortunate emergence of more vaccine-resistant variants as this virus finds its way through the human system. Also, just to make you aware, this being a coronavirus, this is a zoonotic type virus. And so the virus is exploring literally multiple species as we speak. I just heard recently from my colleagues, as being a veterinary virologist, the zoos are experiencing big cat, large primate infections with COVID as well, going from human handlers to the animals. And so the virus is exploring really a lot of antigenic space right now. All right. It's safe to say, I don't want to go through this in great detail, but the infectious disease climate, of course, is unfortunately well and alive. I think the point to be made is that we're finding that many infectious diseases are actually a major contributor of morbidity and mortality, either directly then and or indirectly in ways that we haven't appreciated, where they're finding new linkages between having been infected and subsequent development of additional diseases, or not being able to, in fact, diagnose correctly some of these viral and bacterial and parasitic diseases, and then it leading to additional outcomes, such as a whole new family of the rhinovirus, which is now known to create, and if occurs in infant childhood diseases, develops and exacerbates asthma, COPD, and that complicates things like congenital heart disease, so on and so forth. Examples where bacteria like Porphyromonas gingivalis is now found to be highly associated with systemic inflammation, polyarthritis, arteriosclerosis, et cetera, et cetera. So we, of course, will have a large challenge with meeting these types of pathogens going forward. And the limits of just the acute infection may not be so obvious. Here, I think it's important to note that we are very good at making vaccines which are transmitted in a younger setting generally. For infants, young children or so, childhood vaccination programs have been developed around these. And in many cases, these look to be viral infections which can transmit serially, but then they leave quite a bit of immunity and memory behind. And so as young adults, you don't get really reinfected again. And the infection is that of more of a pediatric setting. Unfortunately, there is a whole group of organisms which don't behave by these rules. And our vaccines are very limited or require remaking annually, such as we see with flu. And unfortunately, we don't have the viral virus vaccine, but as you'll see, and coronavirus, the COVID-19 SARS-CoV-2 may well fall into the flu-like annual type infection. And the cases here will be needing to address variation and mutation, which unfortunately is most likely going to occur. It's also interesting to note that this virus or viruses or pathogens that can explore an ecosystem from in utero, literally from cradle to grave, is a large one. And I think we may be seeing, such as SARS-CoV-2, we have sort of a resistance that occurs in a very young setting, young population. And as we get into an older setting population, certainly here in America, we suffer, as you heard from Mehan, a pretty diverse set of poor health conditions. We're a relatively highly inflamed society from both our nutritional basis and others, environmental and such. And as a consequence to that inflammation that is found systemically is a breeding ground for evolution. And unfortunately, it seems as though COVID-19 has marked people in many aspects to that inflamed state. And so we need to be aware of all of this evolution, and a pathogen which can literally take advantage of a long-lived niche. In other words, be able to continually reinfect hosts over various ages can be somewhat, unfortunately, a very successful pathogen. So I'd like to review quickly class 1 and class 2 pathogens as the term is being used here. You might think about smallpox, mumps, rubella, polio, where we've had incredibly important successes in managing these infections. But they seem to be, of course, an infection followed by a more long-lasting memory and or very few limited future infections. And under those conditions, we're able to, of course, equate that to pretty profound immunity by vaccination. Those pathogens don't have a large degree of capability of evolving and mutating within the host. They have a rather significant restriction on their protein structure. And so they're somewhat limited. They need fresh new hosts to essentially survive in. As opposed to class 2 pathogens, which have decided it would be a much better strategy to be able to reuse the human host over time. And so to accomplish this, you have to solve a couple of problems, none the least of which is on the viral side, being able to underscore and evolve protein mutations, a fair number, and being able to allow that to keep you as a fit pathogen so that you can continue to infect, because certainly as you mutate to escape immunity, you can limit your ability and the threshold for infectivity. And so here we find these class 2 pathogens have evolved an array of immune evading and immune dissuading and diverting activities, which allows them to come in and then in this case, develop a high level of strain restricted immunity, meaning one sort of specific, if you want to call it serotype or fingerprint, is what the immune sees. And then the virus undergoes this drifting and mutation in various locations, and of course can reinfect over a period of time. And there's a long list of both DNA, RNA viruses, bacteria, and parasites that can do this. And it is in this setting that I believe our SARS-CoV-2 may unfortunately end up. And so we need to address these infections as they continue to be the major problem facing both the US and the world today. So a while back, we coined a term and published term deceptive imprinting, which is alive and well today and seems to be an immunologic phenomena whereby the host, both innate and the adaptive immune systems, are either turned off or misdirected during the primary infection. And quite interestingly, from the adaptive side, focuses the immune response on, if you want to call it decoying epitopes, the combination of something being seen, meaning immunodominant, and at the same time not being highly protective. And so in a way, it's sort of raising a flag over here to essentially attract the immune system, while at the same time keeping the immune system from possibly attacking what it is that would really be preventative and to prevent escape and mutation. So I don't have time to go into it, but the development of this is in the literature, and that is that we now find that there are biases. We have immunodominance, which is an important feature of protein and other antigen structures in the body. And that what that means is essentially there are areas or regions of these molecules which have an infinitely more interesting attraction by our immune systems too. And so thinking or being taught that our immune systems and host defenses were sort of this blind, blank slate that was able to be developed against, billions of types of antigens is in fact really not the case. We, as we study this more, we're finding that there are very limited and obvious biases that are built in to the host immune system. And so you've got immunodominance biases of structure, you've got the biased aspect that the immune system plays with, and then you've got the pathogen, which is, of course, in the middle, bridging these two things. All of these contribute to this deceptive imprinting process. So where does it come from? It certainly has something to do with repertoire sculpting. And so you're born with the ontogeny and the phylogeny of your immune system. You have a sculpting that goes on germline and in utero, self versus non-self. You then are colonized by a large 10 times your own cells numbers of bacteria, both on your skin and throughout your body. And so you've got a repertoire sculpting event going on there. Then you have your environment, of course, sculpting that immune system, and then your acquired infections. So all of these give you various biases in public clonotypes and private clonotypes of your B and T cells. And so you already come already set up to some extent. We really don't understand yet all of this, but with the large metagenomics going on in immunogenomics, I think we're going to start to get a much better handle as to how this all works. Now, with that, I'd like to share with you this coronavirus paradox, as I'd like to call it. It really gives, I think, a really good example of how one RNA virus family, that is the coronavirus, which is one of the more successful RNA viruses on the planet, is able to exist in two different forms as a class 1 pathogen and a class 2 pathogen. Polio, as you certainly can recall, was a very important endemic pandemic in the early 50s or so, leading to a couple of vaccines, which turned out to be very important and eliminate, for the most part, those viruses. Well, in that case, poliovirus, as a coronavirus, remember, developed 3 strains in total. There were 1 or 2 that were a little more dominant than the other, but at the end of the day, it had a limited number. Whereas its kissing cousin, human rhinoviruses, may well have over 160 plus strains. And so you can see already there's a complete difference. If you looked at these 2 viruses sitting on the table, you wouldn't know the differences. They're so similar. They're non-enveloped, capsid-like viruses. And so really the question becomes, is what's behind this? How is it that this relatively plastic RNA genome can be both keeping robustness, conservation of structure and function, yet plasticity, so that it can undergo mutation? And of course, how does it outpace our human immune system with such a small RNA genome? And then, of course, how do they conserve this potential? And what are some of the basic principles that we are used to seeing? So if I may then share with you some evolutionary insights where we looked at this with rhinovirus to get a better handle on that. And this was some work that at the time we were doing at Biological Mimetics with a team at DARPA. where we were looking at various basic evolutionary pressures on living systems. And so I'll share with you some of that data. In this case, we were interested in recapitulating an experiment in vitro, and we also had a chance to look at this in humans, given that there was an FDA-approved rhinovirus that was allowed and licensed and had been used and is used and studied in mostly college-age volunteers who received the inoculation and then can undergo sampling and study. So basically, rhinovirus is a capsid virus that has very VP1 through 4 structures. You can see the picture and the description there. Here is a bit of a close-up of the business end of that capsid structure. And above you can see that of the polio virus, and then you can see that of a rhinovirus. And what's interesting is, think of this as a topology. These are like mountaintops, mountain ranges around a large lake, sort of valley. And what you can see with polioviruses, you've got sort of this somewhat triangular set of mountain ranges, and then you've got this lake-like structure. And here in rhinoviruses, you see a similar evolution, but with a bit more peaks and a much deeper canyon and a much narrower lake. And so this is the receptor binding domain for each of these viruses, is the blue-green area. And as opposed to rhinovirus, poliovirus cannot play very much with the structure around this receptor binding domain. It's fairly rigid in that structure. Whereas in this form, where you have higher peaks and a much deeper canyon, there's plasticity around the mutation and evolution that can occur within these 3 regions. And this actually constitutes the evolution. I don't have time to go into this, but work that we published on sharing this and also the same application of this to seasonal influenza demonstrated that these epitopes, these immunodominant decoy regions, actually take turns and oscillate in their dominance over periods of 8 to 10 years within the human population. So what happens is this area here, as you can see, is rather extensive, is a large area that is associated with mutations. And this may carry the virus for anywhere from 5 to 8 to 10 years in variation here, after which mutations here have accumulated. They're too significant, and the threshold for replication are destroyed. And so that virus is no longer competitive. And so it switches, and you now have mutants that have shown up in these additional 2 areas. So you end up with an oscillatory kind of activity. And that's how these pathogens preserve their antigenic relatedness and unrelatedness in the human population or animal population over a long period of time, which is, of course, very important for us to understand, so we can now interpret better how these guys do what they do and how we can circumvent this incredible evolved process. So with that, this is, as we called in the lab when the work was done, sort of the bombing diagram of the canyon and the rims, where we were looking at where these epitopes were and how we could eventually study what happened with them. And so we took a couple of antibodies at the time that were directed to these. They were mouse monoclonals. And we knew that this work had been done earlier, about 20 years earlier, actually, along with the crystallography, where the antibody was put onto the virus, and they looked at its first escape mutations from the antibody. And what was fascinating is we recapitulated that experiment with the exact same reagents, same virus, and asked ourselves essentially what would we see. This is the area of the antibodies that were selected. One was selected, which was a rare and more robust neutralizing protective antibody, and then one which was commonly derived and was a very highly serotype restricted antibody. You can see they sit almost on top of each other with only a slight variation in location. And actually, if you look at them crystallographically, this is an example of how the binding angles, literally 20 or 30 degrees of binding angle and a slight shift in binding around the rim, gives you 2 completely different behaving antibodies, one which is very rare to get and would be more protective, and one which is more common and unfortunately highly strain specific. So we developed the experiment for immune pressure with these antibodies so that we could watch and map and evolve the responses that occurred. At the time we used Human Genome Sciences and the Ventnor Institute to do massive parallel sequencing, and then look at the outcomes that were done. And this was, in a sense, an evolutionary study over time of a sort of time lapse, if you want to consider it, and what happens. We made a massive amount of primers to overlap all these regions. We covered anywhere between 6,000 and 10,000 sequences runs per nucleotide. And so we really tried to cover as much of the landscape as possible. As you'll see here, we are looking at the antibodies, that of 17, and that was compared at 3 days of replication. And it was compared then to a polyclonal antibody, which was raised to the same virus and theoretically has multiple B cell clones, multiple antibody types, being more robust. And of course, then theoretically being able to bind more sites on the virus and inactivate this virus more effectively. And so here are the isolates that were studied, and we focused in on part of a linear sequence, which is found around the rim and canyon that I showed you earlier. And I'm going to share with you the results, actually, which were pretty profound. Where you see the yellow brackets is the data we're going to basically talk about. I want you to look very closely at 3 days of replication. A very specific mutation has already occurred within these epitope domains. What's interesting is that at day 3 days or 6 days, we started here centrally in the VP90 region and then went to these additional sites. And if we compared that to just 3 days of replication with a polyclonal antibody, now where multiple antibody types are available, what you find is the exact same mutation. And what this literally says is that this virus is able to, in its native structure, to induce in a setting of a polyclonal response, a very narrow response. So this would be essentially as collapsing your immune system to look very much like a monoclonal antibody in terms of immunologic pressure. So it says by definition, the natural antigen is able to focus the response regardless of if it was made by the virus or potentially the capsid antigen, thereby giving you this very narrow response. That's real trouble when it comes to developing antigens. And it certainly says that the native antigen itself, which was proven over years of cold vaccine development work, was inadequate by developing high strain restriction. Well, we now know why. And so here are the mutations in red, just to give you an example, show you how identical those were. Well, the story got even more fascinating when we then looked at the original 1985 study that was published. And in there was the exact location and amino acids that we saw as in our response done 25, 30, almost 30 years later. So this suggests, of course, that there turned out to be hotspots within the genome of the virus. And so you've got a high fidelity event going on that's inherent in the system. And of course, has memory. Over time, that same virus and antibody combination yields you the same outcomes. So it certainly is not a random, highly random event by any means. It's a very constructed event and something that we need to better understand, of course, to get a better handle on this. Now, jump over some of the human work, but we studied the same evolution in the infected college volunteers. And we were able then to map all of these insights and understanding on and into the virus. And what was important there is it was our goal to essentially be able to take these immunodominant epitopes away, mutate them in such a way that we could now make them more invisible to the immune system, and yet retain the complex structure that was needed. And then, of course, test that immunogen back on top of the immunity that was compared to the natural analog. And so I'm going to jump ahead here and show you a schematic of the results. Basically, this is what seems to occur under the native conditions. And by various means, by either amino acid substitutions or introducing end-link glycosylation sites where we can, we're able then to with very minimal amino acid changes, make these modifications. And so now, rather than being attracted to these decoy epitopes, which drive strain variation and serotype variation, and of course prolong the evolution of vaccine-resistant mutants, we're now able to mask those. And of course, what we found is that we get a completely new set of antibodies, as you see here in green, that are now focused on the more conserved aspects of the structure and allows the virus to not be able to essentially evolve to any high degree. This approach allows us now to develop antibodies and preventative therapeutics and actually improve diagnostics where monoclonals are inherently used in this process. These are difficult and may limit your ability to identify mutants versus those that are identifying conserved strains. And so this technology called immune refocusing technology allows you then to further develop these immunogens. That has been done with human rhinovirus, and we are in the early development of preclinical, clinical rollout of a first-in-man human rhinovirus vaccine, which we're able to now broaden the immunity from a few serotypes to covering about 62% of the serotypes at this point, with only 3 or 4 amino acid modifications within the capsid. So we're excited by these results and believe this will help set us up for next generation to address the, unfortunately, we believe what will be the ongoing flu epidemics that occur annually, and of course, what may turn out to be a larger COVID type, unfortunately, pandemic or an epidemic that's occurring annually at this point. So with that, I'm going to stop and thank you for your time and answer questions if we have it. If not, enjoy the rest of the meeting.
I request if anybody has a query, can just raise their hand and we'll just call you out. And you can have any doubts if you have with Peter Nara. We have like 2 minutes for a Q&A session here.
Sorry, I can't see the button to raise my hand, but can I ask a question, please?
Yes, Miss.
Yes, please.
So that was a really interesting talk. Thank you. Hang on, I'm just going to put mine. That was really interesting. Thank you. But I was just wondering how, when you introduce the glycosylation sites, is that altering the processing of the antigens? Or different antigens being presented on the MHC class 2 molecules or class 1? Is that what's changing?
Yeah, so that's a very good question. And we don't know all the answers yet to it. We do know that it is in some cases altering that processing. So on the T-dependent side, we know we can make changes and alter that processing. We also know that it is giving us a sort of a conformational shielding of the molecule itself, which is engaging B cells as well. And so we're literally, by using a self-epitope, that of our own glycosylation system, is allowing us to benefit from both T-dependent and probably maybe T-independent processes, which is giving us a little bit more breadth and opportunity to refocus those immune responses that come later. So we believe that it's affecting both T and B cell processes.
Okay.
The only thing I would add is that unfortunately there are not always regions which we can take advantage of introducing, as you might imagine, an NXT or NXS site. But where we can, particularly with flu, we found in the envelope with the HA protein, we only need to make 2 or 3 glycosylation sites. They're not always utilized, but in many times they are, because in fact, we found that viruses that escape immunity actually introduce glycosylation sites themselves to escape the immune system of the body. So they're literally doing it against us as we do it against them.
Yeah, that's interesting. We should probably talk separate to this at some point, because we're working on a new system for trying to introduce glycosylation sites into proteins. So
oh, sure, please follow up. Yeah, very much so. Yes, thank you.